So guys, lipids not only occur as only lipid molecules, they also tend to conjugate themselves. And one of the very important conjugated molecules are those one when lipids are linked with the proteins to make lipoproteins. The lipoproteins, of course, are very diverse compounds, but the question should arise in your mind that why lipids have to bind with the proteins. It very much lies in the very structural nature of the lipids. You know that lipids are hydrophobic in nature, and of course this gives rise to many functional aspects to lipids also, but it also gives some challenges. The challenge is that the whole cellular environment, is aqueous. The body environment is aqueous, and the lipids have to move from one part of the body to the other for digestion, for giving rise to energy, for fulfilling whatever function, whatever body needs. For that reason, the lipid have to bind with the protein so that it can move from one part of the body to the other, or one part of the cell to the other part. Let's see that what are the different types of lipoproteins, how they are synthesized and metabolized, how they are transported from one region of the body to the other, and what is its significance. The composition of lipoproteins is that they are arranged in a specific manner. The different lipids are arranged in a specific manner. For instance, the core of this molecule contains the hydrophobic compounds like the esterified cholesterol and triacylglycerol. So they both form the hydrophobic core of this compound of the lipoproteins. Then this core is surrounded by the amphiphilic apolipoproteins, phospholipids, and free cholesterol. This molecule, while it's moving, it's oriented in a way that it can easily float through the aqueous environment. The sources of triacylglycerol and cholesterol, you know that it comes from the diet which we eat on daily basis, and also on to the de novo synthesis or endogenous synthesis. Then there are different classes of or types of apolipoproteins, and we categorize them on the basis of the composition of the lipid they contain and the protein they contain, because more the lipid this apolipoprotein will have, lesser will be its density, its size will be higher. So as a result, the least dense of the apolipoprotein are the chylomicrons. They are the largest and the least dense, and also because they are very large and least dense, it's because of the reason that they have highest amount of triacylglycerol, and of course they will have the lowest protein content. Then there come the VLDL and LDL, very low density lipoproteins and the low density lipoproteins. They are of course denser than the chylomicrons, but they are still are not not as dense as the HDLs we will shortly see. The HDL stands for high density lipoprotein. They are smallest in size and densest also. Why they are smallest? Of course, because the lipid content is much less and the protein content is much more in them. They can be separated by different methods. We will not go into the detail of this because this is not really the focus of our presentation of our lecture. Then as far as the roles of apolipoproteins are concerned, many very diverse type of roles it does. But one of the most important role I have already told you, that they move lipids from one part to the other. In addition to this, they also act as the recognition sites for cell surface receptors, because they have proteins, and those proteins then act as a signaling molecules which can act as receptors also, or binding with the receptors, act as activators in lipoprotein metabolisms. It leads to the metabolism or contributes towards metabolism. It have many structural components also, and these structural components, like different lipoproteins it contains, apolipoproteins it contains. The proteins on the lipoprotein is known as apolipoprotein. They have ApoB, ApoA, ApoC, ApoE, and so on and so forth, and they give not only the distinctive characteristics to the lipoprotein, but also functionally the functional differentiation also occur because of this different amount of or different types of apolipoproteins which are present on the lipoprotein. Because there are so many types of apolipoproteins, and as a result there are so many types of lipoproteins, we know very much about them, but still there are many areas which are unknown, especially how different combination of these apolipoproteins perform differently. What are those different functions? We don't know. We know to some extent, but still there are many gray areas because of the diversity they have. Because the heterogeneity these molecules have.